of prokaryotes, but they also have similar vulnerability to antibiotics, particularly antibiotics. And the thing that really clinched it is the genomes. So if you look inside of mitochondria and inside of chloroplasts, for example, they have their own genomes, completely distinct from the chromosomes of the nucleus. And the genomes of mitochondria and plastid are circular, double-stranded molecules, like those of prokaryotes, of bacteria in particular. And if you sequence the DNA of these organelles and put them in a phylogenetic analysis that includes bacteria, archaea, eukaryotes, they come out as most closely related, in the case of mitochondria, to proteobacteria, and in the case of chloroplast DNA, to cyanobacteria, the blue-green bacteria that are photosynthetic. So there's really no controversy about this anymore. It's pretty well nailed down that the endosymbiotic theory explains the presence of mitochondria and plastids in eukaryotic cells. There's another level of this endosymbiosis, though, that's really pretty fascinating, and that's what we call secondary endosymbiosis. And this accounts for the presence of plastids in a lot of these algal lineages we're going to talk about here. So the idea why it's called secondary endosymbiosis is this is an instance where we have to hypothesize that an herbivorous, non-photosynthetic, eukaryotic unicellular organism like the one shown here in orange ingested not a blue-green bacteria, not a cyanobacterium, but a unicellular eukaryotic alga that already descended from an ancestor that had ingested a cyanobacterium and had, through evolutionary time, evolved plastids from that bacterium. So here you see a eukaryote with a chloroplast here. This is not a blue-green bacteria being ingested and eventually totally incorporated into that cell. So that's some of the evidence that this has occurred, is that there are extra membranes around these plastids, these chloroplasts, in some of these algal lineages, more than you need to explain the two membranes that surround the bacterial organism. And these represent the membrane of the plasma membrane, the cell membrane of the eukaryote that was ingested in part. So that's something quite interesting, and there's some good evidence for this. So here we have a little diagram that's really a nice little heuristic to study. That's also in your book. That shows first this primary endosymbiotic event, then, where we have a blue-green bacterium, also called cyanobacterium, captured by this heterotrophic unicellular eukaryote that already had mitochondria. Mitochondria came first. And then we had evolutionary divergence right here. This is just natural evolutionary divergence between, there's diversification here, resulting in a group called the green algae and a group called the red algae, which have also diversified a lot. But early on, one of the green algae, they're unicellular, and some of the unicellular red algae were ingested by different eukaryotes that were non-photosynthetic, and we had incorporation of these organisms into the cells to form the plastids of these organisms out here at the right. So there are three different events at minimum that represent secondary endosymbioses that account for modern-day photosynthetic organisms. And again, this accounts for all of these disparate lineages here having plastids. It's because they've independently acquired them through secondary endosymbiosis. It's not that they evolved photosynthesis independently. They basically borrowed photosynthesis or co-opted it from organisms that had acquired it earlier, and everyone ultimately acquired it from the cyanobacteria. And if you go back far enough, those are the only organisms that evolved photosynthesis or oxygen-generating plant-like photosynthesis de novo that are still around today, and that account for all of these examples of photosynthesis. So if we look at the algae, then it's not surprising we have a lot of diversity. And we often hear algae broken up into two major groups. Phytoplankton, which is basically the plankton composed of so-called plants, which is what phyto refers to. And plankton are just organisms that are out in microorganisms, unicellular organisms usually, that are suspended in water bodies like upper layers of lakes in the ocean. And you can see there's a wide diversity of these phytoplankton groups, the blue-green bacteria, and then these groups that represent secondary endosymbiotic acquisition of photosynthesis. And then there are multicellular groups called seaweeds, oftentimes, of course, only if they grow in the ocean, but brown algae, red algae, and green algae. And these have all acquired multicellularity independently of one another, these three groups here, and are unicellular members of these two groups that are still around, the red algae and the green algae. So let's have a look at the blue-green bacteria again first. Remember, we saw these when we were talking about prokaryotes in the first lecture. So the cyanobacteria are unicellular, but we do get some taxa species that can grow as loose filaments, and they can even have specialized cells within these filaments that are responsible for nitrogen fixation. So we'll talk about that a bit. Gaseous nitrogen is abundant in our atmosphere, but it's not available to plants. But cyanobacteria and some other bacteria in the proteobacteria group can make it available through various chemical reactions. So this is a really important group, both for oxygen generation, nitrogen fixation, and also as the original source of the plastids in organisms that are photosynthetic. So among these groups that acquired photosynthesis through secondary endosymbioses, which are the groups out here, these are the groups that acquired it through primary endosymbiosis here, the red algae and green algae. Among these groups, the chlorarachnophytes is a little group we're not going to have much to say about. It represents one of those major lineages of four major lineages of eukaryotes that are photosynthetic. But it's a unicellular tropical group in tropical oceans. And the interesting thing about it is that there's actually a residual nucleus inside the chloroplast. You don't expect a nucleus inside of a chloroplast, but that's evidence this was a secondary endosymbiosis. Absolutely ironclad evidence. There's even some DNA inside that little nucleus, which we call a nucleomorph. And if you sequence that DNA, it shows that it's closely related to algae. The plastid is, the green algae, I should say. So it's clearly a secondary endosymbiont. There was another independent secondary endosymbiotic event that involved a green alga, and this gave rise to the euglenids. The euglenids are a really diverse group, pretty common group, pretty ubiquitous, that are green, and there's a good reason they're green. They've acquired their plastids from unicellular green algae, but they store their starch in an unusual form. They store their energy in an unusual form, not as starch, and they're capable of phagocytosis, of actually engulfing prey items, just like the hypothetical ancestor that engulfed the original endosymbionts. And so this is an organism that still shows the ability to do what it must have had to do in order to acquire that green algal endosymbiont that became its plastids. And some of these are heterotrophic. They can lose their chloroplasts and still be capable of survival by engulfing other organisms. And even some of those that have the ability to photosynthesize also will occasionally snack on other organisms. So they're what we call mixotrophs. They can be heterotrophs or autotrophs or both. All right, and the last major clade here that's an example of secondary endosymbiosis is one that acquired photosynthesis by capturing a unicellular red alga, another eukaryotic group. And this is a hugely diverse clade of photosynthetic organisms and some organisms that have lost the ability to photosynthesize secondarily. This is a particularly interesting one. I'm not going to have much to say about it, except that this group called the apicomplexans includes Plasmodium, which is the organism, not to be confused with the plasmodial state of slime mold. I'm talking about the genus Plasmodium, which is the organism responsible for malaria, which kills millions of people every year, one of the worst organisms in the world for human health. And it actually has a vestigial, rudimentary, little non-functional plastid inside of its cells, which might be its Achilles heel in the sense that some medical researchers are trying to target that plastid as a possible site of vulnerability of the organism. So it may still be important that it has a plastid for human health outcomes in the long run. But these other two groups here still are photosynthetic as a result of this event. And we'll talk first about the dinoflagellates. So the dinoflagellates are a very bizarre group, a really cool group that has a really strange, even though they're unicellular, they have a really strange morphology. Here you can see some of these things. They have bizarre projections and plates of armor. They're really interesting to look at microscopically. And interestingly, these plates, this outer cell wall covering is made of cellulose, just like the cell walls of higher plants, but this is an independent case of, it's an example of convergent evolution. And an interesting thing too, they have a couple of flagellae, but one of those flagellae actually circles around the body in a groove, and it moves in a way that causes the organism to spin through the water like a top. So they have a really different way of movement or a different form of movement than other algae. And you probably heard about them because they form major blooms at times that are responsible for what are called red tides. And here you can see a red tide, this orange coloration near the shore here, which is basically just a huge number of dinoflagellates that have the coloration is from the carotenoid pigments in their cells that causes this reddish coloration. And an important thing about their biology is that they use toxins to defend themselves and also to paralyze their prey. So in addition to being photosynthetic, they also have the ability to be heterotrophic as a group. And they, when they're in these heavy concentrations, these toxins can kill fish, and they can render shellfish, mollusks, poisonous to humans. So they're sometimes, this is really important to keep track of dinoflagellate activity, and it can have major importance to harvesting of food from the ocean. But one of the most important ecological, well, there are a couple of really important things about them ecologically. For one thing, they're incredibly abundant, and not just when they're in red tides like this, but they're a big part of our phytoplankton, and they're responsible for a lot of the oxygen in our atmosphere. They're also important for fixing a lot of the carbon and reducing the amount of carbon dioxide in the atmosphere. So they're important allies in the fight against greenhouse effect. And they also are symbionts, some of them with corals. So they actually live inside of corals and are important for the survival of corals, which of course are fundamental to coral reef ecosystems, which are some of the most diverse marine ecosystems. All right, so finally we're going to talk about the straminopiles, this other major clade, and that just means this straminophile word refers to this this feathery or splumose flagellum that they possess as a group. And that's not so important. It's not important at all, but that's, if you're curious, the basis of the name. And the straminopiles include a few really important groups of algae that have all acquired their ability to photosynthesize from that same incorporation of a red alga into their cells and shared with the dinoflagellates. One of the most important are the diatoms, which are spectacularly beautiful and diverse organisms. These are microscopic unicellular organisms, occasionally in colonies. And these intricate cell wall patterns are really important to these organisms' survival. They increase the strength of their cell walls. And these cell walls, this might look like a petri dish, and they really do look like little petri dishes. Their cell walls are made of hydrated silica, so basically glass-like cell walls. And the two halves of the cell walls are in two halves that fit together like the two halves of a petri dish. So it's quite a strange organism, extremely diverse, and they account for about 25% of the primary productivity on Earth from photosynthesis. So they're really important not only for as autotrophs, basically capturing carbon and making carbon available to other organisms as food, but also when they die, they're suspended in water bodies, like in the upper layers of the ocean and freshwater lakes. They're actually really diverse in freshwater lakes as well. When they die, they sink rapidly, and because of that, they leave the ecosystem. Basically, the carbon that they've captured is taken out of the carbon cycle, and they're basically removing carbon on a mass scale. So these are really important organisms for fighting CO2 buildup in the atmosphere. And some people have proposed seeding the oceans with iron to try to promote their success and to capture more carbon dioxide and remove it from the ecosystem. But there might be unintended consequences, so there's a lot of worry about trying to do that, but it is something that's been proposed. All right, now the reproduction of an organism with glass-like cell walls is a little bit of a challenge. These are not expanded. They can't expand at all, these cell walls. So what happens, these organisms, when they reproduce during cell division, there's a new half of each, so the cell splits apart, and a new half is formed within the old half. And so if you keep that up over time, you're going to have some lineages that are going to continue to get smaller and smaller as each cell division results in a new cell wall being formed inside the old one. And at some point, diatoms will become so small that they couldn't continue to exist. But what happens is when they reach a certain minimum size threshold, then they have sexual reproduction. This is all asexual reproduction that's going on here, basically cloning of the cells. But then when they get below a certain size, that stimulates meiosis. We have production of gametes from meiosis, and then ultimately the production of a zygote after fertilization. And then the new organism grows to its full original size before the cell walls form. So we're back up to the original size again. So that's a pretty cool way that these guys reproduce. And they have a life cycle like ours. So the diploid, the only haploid phase are the gametes. And they're actually closely related to the Mycota, remember those water molds that are responsible for tree diseases that aren't really fungi. These are among their closest relatives, and they also, as I mentioned, have this kind of life cycle like ours. Now, the golden algae are another straminophile group. I don't have much to say about them. They're golden because of these carotenoid pigments. But they're usually unicellular, occasionally colonial, like here, mostly planktonic organisms, both in freshwater and marine situations. But it is a distinct clade of algae from that same ancestor. And one of the most interesting groups of straminophiles, and by far the most elaborate, are the brown algae. And again, brown rather than green because of carotenoid pigments. But these can get to be huge, and they can look very tree-like. This is an organism called the sea palm, and you can see why it's called the sea palm, because these look like leaves, these blades. The stipe looks like a stem, and the holdfast that affixes it to the bottom, or to a substrate, looks like roots. But this is completely independent of land plants. These have an independent origin of multicellularity from a unicellular ancestor. And this includes a lot of our seaweeds and kelps. Here you can see some kelp forest out in, well, off the coast here. These are really fundamental to the ecosystem, basically keystone species. And we'll talk a little bit more about their biology on Monday. And then we'll get into land plants right away on Monday. All right, so have a great weekend.